The overall goal of the study is to implement whole genome sequencing for identification of mutations in genes that confer antifungal drug resistance in Candida glabrata in a diagnostic laboratory.
Whole genome sequencing of fungi can make significant contributions in the field of antifungal drug resistance. It allows simultaneous detection of genome-wide mutations in drug-resistant fungi to important antifungal agents.
Whole genome sequencing of medically important fungi can also enable the detection and monitoring of outbreaks of fungal disease and transmission events in healthcare settings, as well as emergence of fungal species and clones that clinicians would need to know about.
Demonstrating this procedure will be Dr. Rebecca Rockett and Dr. Verlaine, postdoctoral research fellows from our laboratory.
To begin, prepare genomic DNA samples from clinical and commercial strains of Candida glabrata. Then, add 5 microliters of quantified DNA to each well of previously labeled fresh 96 well, hard shell, thin wall plate. Add 10 microliters of tagmentation buffer and 5 microliters of amplification buffer to each well. Then, use a pipette to gently mix the samples and seal the plate with an adhesive seal. Put the plate inside a thermal cycler and run the appropriate PCR program. When the sample has reached 10 degrees